speakers. I will refer to their contributions, which were significant during this talk. What I'm interested in today is how the evolution of the mode of female provisioning of offspring affects the evolution of sexual antagonism, and in an indirect sense, also the rate of speciation. I'll first talk about the natural history of the organisms I study. I'm working on the fish family Poeciliidae, and what you're looking at here are samples of males in 4 species in the family. And they represent what the family is best known for, which is species that are subject to extreme sexual selection, which has been the main subject of study. What's less well known and illustrated in all these pictures of either individual males or males and females representing 14 genera in the family is that in most species there's nothing special about the males. I'm also interested in female variation, and in this case I'm interested in how mothers provision their young. All species in the family, save one, bear live young. Almost all the species in this family are really egg retainers, and what that means is that they fully provision eggs before they're fertilized, and then they contain them inside through development and give birth to live young. What I discovered in 1987 was that there are papers written in the 1930s and the 1940s that showed that some of these species have the functional equivalent of a mammalian placenta. And here's an illustration of what those features look like in 2 of the species. The other thing I discovered was that even then, it was clear that the placenta had evolved more than once. It wasn't the same in each of them. And what was more amazing is that 40 years later, when I was reading about this, almost nothing had been done to further explore the diversity of how these animals provision their young. And so for me, it became a hobby to travel, collect fish, go to museums, and characterize the mode of maternal provisioning. And 20 years later, in collaboration with Robert Meredith and Mark Springer, we were able to produce this. This is a well-resolved phylogeny for the family, and what we've done is to map on this phylogeny the mode of female reproduction. We have multiple variables that we looked at, but there are 9 arrows here that represent independent origins of placentas. The placenta had evolved independently at least 9 times within the family. This is a visualization of what you see when you dissect these fish. These are embryos early in development, and these are embryos advanced in development and getting close to birth. This number, the matotrophy index, is simply the ratio of the dry mass of the embryo at birth divided by the dry mass of the egg at fertilization. And so this species has about an 8-fold increase in dry mass during development. This one has about an 85-fold increase. The most common value that you see is about 0.7, meaning that the embryos are actually losing weight during the course of development. 0.7 is about what you would see in a bird egg, which is why we infer that the eggs are fully provisioned before they were fertilized. Bart Pollux assembled a comparative data set on males, and here I illustrate 3 of the properties of males that he evaluated. The first is the presence or absence of courtship. This is a male sailfin molly courting a female who's in the back. The alternative is what we call covert copulation. This is a male that's just sneaking up behind the female and trying to inseminate her. So this is a trait that's either present or absent. Sexual dichromatism is also either present or absent. In the male sailfin molly, there's iridescence on the dorsal fin and the caudal fin, and then orange pigmentation on the body. The female is a uniform silver gray. In this case, the sexual dichromatism is absent. The male and the female have the same color pattern. A third trait is ornamentation. This male is ornamented in having an enlarged dorsal fin and enlarged tail, plus he's laterally compressed. And the nature of the ornamentation reinforces the courtship behavior, and this male lacks ornamentation. Another trait right here, highlighted with the white line, is the length of the gonopodium, which varies remarkably from about 15 to 50%. This genus is Phyllicthes, which translates roughly into English as dickfish. In summary, when we did the ancestral reconstruction of these traits, what we found was that ancestral females lacked placentas. There are at least 9 origins of placentas. Once gained, they're never lost. The ancestral males lacked all traits associated with sexual selection, and there have been multiple gains and losses of these traits throughout the history of the family. Okay, what I'm interested in now is a hypothesis proposed by Zet and Zet called the viviparity-driven conflict hypothesis. And what they did was to consider what the consequences might be in the evolutionary shift from providing all resources to the offspring before fertilization to providing most of those resources to offspring after fertilization. What they had predicted is that this shift in maternal provisioning should be associated with a shift from pre-copulatory mate choice to post-copulatory, pre-copulatory mate choice to post-copulatory mate choice. The rationale is that if a mother has invested everything she's ever going to invest in her offspring before the egg's fertilized, if she wants to influence who the father of the offspring will be, the best way to do that is to choose who to mate with, which sets the stage for sexual selection. If most of those resources are going in after fertilization, then they predicted that that would be associated with a tendency to mate with many males, so there are diversity of sperm present to either compete or to be chosen at the interface between the sperm and the egg, or to be chosen after the egg is fertilized and the paternal genome is being expressed. We originally addressed this hypothesis by just looking for the simple association between male traits and female mode of reproduction. This result was published a long time ago. What we found is that the pattern supported this prediction, that the placental species had males with less sexual dichromatism, no male ornamentation, and no courtship. What we're doing here is trying to push this comparison further. We did it first with an analysis that asks whether or not the evolution of a trait in males is contingent upon what the condition of females are, or the other way around. Here you're looking at one such analysis where we're looking at dichromatism and placenta. So you have 2 male traits, 2 female traits, and 2 by 2 means that there are 4 possible conditions. And what this analysis shows is that the evolution of dichromatism, either the gain or the loss, is contingent upon the female lacking a placenta. Here you're looking at the 3 analyses that we did, and the heavy blue lines say that there are 5 of the 6 possible transitions in males are contingent upon females lacking placentas. There's only 1 transition in females that's contingent upon a male trait. We also looked directly at the tree and mapped out where these transitions happened and asked, in the case of males, if you see a derived male trait, what was the condition of the female? And what we found is that derived male traits, traits associated with sexual selection, only originate in lineages that lack placentas. Placentas could originate in lineages where the male is predicted to either have or not have traits that are associated with sexual selection. And those that did have them, in 2 out of 3, those traits tended to be lost after the placenta evolved. And so the trend overall is that there's strong support for the mode of maternal provisioning governing the evolution of male traits, as opposed to the other way around. Zet and Zet made a second prediction, which is that the evolution of the placenta should cause an accelerated rate of evolution of post-copulatory reproductive isolation. And there's some empirical data in support of that idea. And they further predicted that because of this, placental species should have higher rates of speciation. So we can address whether or not that's true. But we have an interesting contrast here, which is that we also have some lineages with highly developed sexual selection. And independently, people have suggested that that would cause accelerated pre-copulatory reproductive isolation and should cause accelerated speciation. So we have a nice situation where you can compare the two. And in this field of speciation, people discuss whether or not pre- or post-copulatory reproductive isolation is playing a more important role in determining the rate of speciation. Here you're looking at the results of analyses that were done in Diversity, but we also did these analyses with 2 other programs that are more conservative, and we got the same results. So these are robust results. But in terms of females, what we found was that the non-placental lineages have higher rates of speciation than placental lineages. So the Zet and Zet prediction is not supported. Here we look at it in terms of males, and we scored males as having values of 0 through 3. 0 means that none of those 3 traits are present. 3 means that they're all present. What you find is that you go from 0 to 1 to 2 to 3, that there's a progressive increase in the rate of speciation. and that those lineages with all three traits are significantly faster in speciation rate than those that lack all three. So the conclusion then is that placental lineages tend to have slower rates of speciation, but those lineages that have the highly elaborate males have higher rates of speciation. But the male traits and the female mode of reproduction are well correlated with one another. And so the question is, can we separate the two? And with diversity you can, because you can set up models that include both the male and female traits, then you can get estimates of the contribution of each of them. And what we show for 3 different analyses for 3 different male traits is that the female trait in pink always broadly overlaps 0 and is not significant. The male trait is always positive and doesn't overlap 0, so it is significant. So it appears that it's the male traits that are associated with the higher rates of speciation, not the female traits. And what this suggests then is that perhaps sexual selection really does accelerate the rate of speciation, as has been seen in some groups, but it's been looked for and not seen in other groups. And it also suggests that pre-copulatory reproductive isolation might perhaps dominate post-copulatory reproductive isolation in governing the rate of speciation in this particular group of animals. So the big conclusion then is that the evolution of the mode of female allocation of resources to reproduction seems to have, as Zet and Zeb predicted, and as I was kind of skeptical of at first, but they gave me hypotheses I could test, so they gave me traction. But it appears that they were right. These associations are there, and in an indirect sense, the evolution of female traits also seems to influence the rate of speciation. Now, I consider this kind of result to be a beginning, not an end. What this result tells me is that there's something compelling going on that's worthy of further investigation. I'm pleased now to say 2 things. One is that these results this paper was accepted in Nature Communications a few days ago, so you can look for it. Andrew Furness is the senior author. He's also the one who did all of these comparative analyses that I've just described. And then the second thing is that we have NSF support to do empirical studies that test more specific hypotheses that were inspired by Zet and Zeb's more general hypothesis. And I hope to return a year from now or 2 years from now and tell you what the results of